Good morning, everyone, and thank you for being here today. I am pleased to welcome you to this presentation on a very important topic. Today, we will be discussing the incorporation of recovery processes in active pharmaceutical ingredients, or APIs, and how this can impact their shelf life. Over the next few minutes, I will walk you through the key concepts, the significance of these recovery processes, and what it means for the stability and longevity of pharmaceutical products. Let's get started. Let's start by talking about APIs, which are the core therapeutic components of drug products. These APIs are typically produced through complex chemical or biotechnological processes, making their manufacture quite intricate. Because of this complexity, APIs are highly sensitive to certain factors. For example, impurities and residual solvents can impact their quality, and various degradation pathways can cause them to break down or lose potency over time. It's important to recognize that recovery processes, how we extract and purify APIs, can greatly influence their stability and shelf life. Small changes or inefficiencies in these processes can lead to significant effects on the API's longevity and effectiveness. Understanding these points is essential because they tie directly into ensuring drug safety and efficacy, which is at the heart of our overall discussion on pharmaceutical quality and manufacturing excellence. Let's take a closer look at what we mean by recovery processes. These are the methods we use to recover and reuse materials during production, which not only helps with efficiency, but also with compliance. Reprocessing is one key aspect. This involves repeating steps like crystallization or drying to ensure we get the desired quality. Reworking, on the other hand, includes additional purification steps to improve purity or remove impurities. Another important element is solvent recovery and reuse. By recovering solvents, we reduce waste and environmental impact while also saving costs. Similarly, recovering APIs from intermediates allows us to maximize yield and minimize waste. Now, from a regulatory perspective, it's essential that these recovery processes do not adversely affect the final product's quality. This means no change in purity, strength, or stability. Everything must remain consistent and within specifications. Overall, these recovery methods are vital for maintaining product quality, regulatory compliance, and sustainability throughout our manufacturing process. Let's now discuss some of the stability concerns we often encounter with pharmaceuticals. First, chemical degradation can happen through processes like oxidation, where the drug reacts with oxygen, or hydrolysis, which involves reaction with water. These reactions can compromise the drug's effectiveness over time. On the physical side, we see changes such as polymorphic transformation, where the drug changes its crystalline form, potentially affecting its stability and bioavailability. Hygroscopicity is another physical change. This is when a substance absorbs moisture from the environment, which can lead to further stability issues. It's also important to note that recovery processes, like purification or reprocessing, may sometimes accelerate these changes, so we need to be cautious with handling and storage. Now, focusing on critical quality attributes, or CQAs, these are key parameters we monitor for APIs, or active pharmaceutical ingredients. These include the assay, which tells us the purity percentage, the impurity profile to identify any unwanted substances, residual solvents, which are remnants from manufacturing, the polymorphic form, since different forms can have different stability profiles, and particle size distribution, which can influence dissolution and stability. All of these attributes directly impact the stability and shelf life of the drug. Keeping these CQAs in check helps ensure that our pharmaceuticals remain safe and effective throughout their intended shelf life. Now, let's take a closer look at the mechanisms of impact. What I mean by this is how exactly our actions and strategies create change. First, we consider the direct effects. These are the immediate outcomes that happen when we implement a new initiative or policy. Then, there are indirect effects, which might be less obvious at first, but can influence broader systems or communities over time. It's also important to recognize feedback loops. where the results of our actions inform and shape future decisions, creating a cycle of continuous improvement. Throughout this process, understanding these mechanisms helps us to optimize our efforts and ensure that our impact is both meaningful and sustainable. Connecting this back to our overall theme, grasping these mechanisms is key to designing effective solutions that truly make a difference. Let's take a closer look at the impact of residual solvents. As you can see from this example, before recovery, the solvent level was around 200 parts per million, but after reusing the solvent, it increased to 800 ppm. This is significant because we must meet the limits set by ICHQ 3C to ensure safety and quality. Higher solvent levels can affect the stability of the product and increase the risk of degradation over time. To illustrate this, we have a calculation comparing the shelf life before and after recovery. Initially, with a low degradation rate constant of 0.00 1 month to the -1, the product's shelf life was about 105 months. But after the solvent was reused, and the degradation rate increased to 0.004 month to the -1, the shelf life dropped dramatically to just over 26 months. So, reprocessing can really cut down the product's stability window. Now, let's look at impurity levels as another example. The impurity limit according to ICHQ-3A is less than or equal to 0.2%. Before recovery, the impurity level was only 0.08%, which is comfortably below the limit. However, after recovery, it rose to 0.18%, which is much closer to the maximum allowed. This reduces the margin for stability and highlights the importance of controlling these residuals. Overall, these examples underscore how residual solvents and impurities can significantly impact product stability and shelf life, making it essential to carefully manage and monitor these levels throughout the manufacturing process. Let's take a closer look at the stability study design. According to ICH guidelines, stability testing is conducted under different conditions to simulate real-world storage and accelerated scenarios. For long-term stability, we typically use 25 degrees C with 60% relative humidity. This helps us understand how the product behaves over time under normal storage conditions. In contrast, accelerated stability testing involves higher temperature and humidity, specifically 40 degrees C and 75% RH. This allows us to predict the shelf life more quickly by observing how the product degrades under harsher conditions. During these studies, we evaluate several critical parameters. First is the assay, which measures the potency or active ingredient content to ensure it remains within specified limits. Then, we look at impurities, both formation of new impurities and the potential degradation products. Physical form is also key. Things like crystallinity, particle size, or any physical changes that might affect stability or bioavailability. Finally, it's important to compare the recovered API, the active pharmaceutical ingredient, after storage to the original API. This comparison helps us assess how much of the active ingredient has been retained and whether any degradation has occurred over time. Overall, this comprehensive approach ensures we thoroughly understand the stability profile of the product, which is crucial for ensuring safety, efficacy, and quality throughout its shelf life. Let's take a look at a hypothetical case study involving an antihypertensive drug. This example will help us see how our API can be applied in a real-world scenario. We'll examine the key points, such as how the drug interacts within the body, its efficacy, and potential side effects. By doing this, I want to highlight the importance of understanding drug mechanisms and patient response. This case study ties into our overall theme of improving personalized medicine and optimizing treatment strategies. So, as we go through this, keep in mind how these concepts translate into better clinical decision-making and patient outcomes. Let's talk about the regulatory considerations we need to keep in mind during recovery. Essentially, recovery is acceptable only if certain conditions are met. First, the impurity profile must remain unchanged, meaning no new impurities should form, and the existing impurities should stay consistent. Second, There should be no polymorphic transformation, as changes in crystal form can affect drug stability and efficacy. Third, the stability of the drug must be maintained throughout the process. If these conditions aren't met, there could be serious consequences. For example, we might face a reduction in shelf life, which impacts how long the product stays effective and safe. Additionally, the batch could be rejected altogether, which means starting the process over and losing valuable time and resources. So the key takeaway here is that maintaining these parameters is crucial, not only for regulatory approval, but also for ensuring the safety and effectiveness of our product. It ties directly into our overall goal of delivering high-quality, reliable pharmaceuticals. In conclusion, it's important to remember that APIs are highly sensitive to process variations. Even small changes during recovery processes can introduce risks, such as impurities or stability issues that could compromise the final product. That's why any recovery step must be carefully managed and supported by solid, data-driven evidence. Stability studies play a crucial role here. They help us ensure that the API remains stable and maintains its intended shelf life. Ultimately, recovery processes are acceptable only when they do not affect the quality, purity, or shelf life of the API, and when they remain within regulatory compliance. It's all about balancing the need for recovery with the assurance that the product's integrity and safety are preserved throughout the process. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional resources you might need. Remember, your success is our priority, and we're happy to assist you along the way. Thank you so much for your time and attention today.